Hello. Today I want to talk a little bit about the Galapagos Genetic Barcode, which, in summary, are two major aspects of my work that merge during the pandemic in a very interesting way on the Galapagos Islands: science and community. So the motivation behind this project is neatly captured in this quote by Margaret Whitley, where there's no power for change greater than a community discovering what it cares about. And this is particularly clear on the Galapagos. The community of the Galapagos depends on their ecosystem. It is a top tourist destination in the planet, where more than 85% of the population depends on these revenues. And unfortunately, during the pandemic, these number of tourists plummeted, and with that was revealed the fragility of industry and the funding that was usually put forward for conservation programs and restoration. So the group of inhabitants that were mostly hit during the pandemic were your naturalist guides, like this person here in front of tourists talking about the sea lion, farmers and fishermen providing all the goods for this industry. And so when you have a population that depends on tourism, there were a lot of new ideas that this community was putting forward to mitigate this economical stress. And some of them were the implementation of negative practices of fishing, like long lining, so they can increase the revenue, or open skies, which was allowing tourists to come from different parts of the globe without going through the mitigation protocols and fumigation that were in place at the airports in the mainland, which were shut down. If that would have been allowed, that means that these planes would have come with potential invasive species threatening the islands. The other thing that we saw that happened is the increase in illegal activities like trading and the poaching of species like these Galapagos giant tortoise babies. They can go for thousands of dollars in illegal markets. So again, the lack of funding from tourists really let the islands out to these illegal and not very environmentally sense practices. So we're going to watch here a testimony of naturalist guides.
So this is when people were in lockdown, as we said.
Yes. Before pandemic, this was not here. I remember, I can say that because I was one of the first guides that arrived to this beach after pandemic, right? The first group, I was one of the ones that visited this place. I was pretty fresh, right? Not like this. Was with the shell was still with a little bit color, right? So I was completely sure that we was doing the pandemic.
Wow. Okay.
So yes.
So there was no control, nobody patrolling here.
No one controlling this. So that is why it's my opinion that the pandemic helps a lot the wildlife in the world, but in Galapagos it's a mess. I know a lot of colleagues, like scuba divers, that went for this lobster for sale. It was not lobster season, and they were selling lobster, right? Because nobody controlled, they have this excuse they didn't survive, and they were taking more than they need from the ocean.
They make money whenever they could, right?
Exactly. So people ask me, okay, so for the Galapagos, good the pandemic, due to the lockdown? It's like, not at all, because we really kind of try to conserve our island, right? We have the national park, we protect, we have the Galapagos rules, but for Galapagos, in fact, we were having these kind of things, not good.
So what we tried to do with this project was to help some of our friends and colleagues on the islands. So the first thing that we wanted to do was to create new employment opportunities during the pandemic. And in that goal, we tried to create new jobs, and we had to provide new skills for the locals and really create a community that is more resilient to future perturbations. So this is the largest citizen science project implemented on the islands thus far. And I think one of the ways and the reasons why this project is such a success on the islands is because we do have a technology that could be implemented and help us reach the scientific goals that we set up. One of the things that we achieve is that 100% of the people employed are from Galapagos. 100% of the data generating, it was done on the islands, and all the information was put forward to the preservations of these islands. So this is something in which, to me, is one of the best examples of how nanopore technologies, it's a vessel to democratizing science and allowing new communities to start being the actors in their conservation efforts. So the Galapagos Genetic Barcode has 6 pillars, and some of them take into consideration the developmental sustainable goals by the UN. And the first one that we needed to tackle was immediate employment. So through our funding agencies, we were able to bring money and employ locals during the time of the pandemic. These are very unusual images of the islands. Usually, these are beaches that are packed with tourists. The businesses are thriving, vendors on the streets, tourists moving from islands on these vessels. And this was in April of 2020. So what we did was to employ directly 74 people on 3 different islands. And their backgrounds were very diverse, including people working on the fishing industry, agricultural industry, naturalist guides, among others. And the participants chose to be part of different activities, like field assistants, lab assistants, leaders of the group. And we tried to have an equal representation in gender in our project. Now, because of the nature of our project, we had a lot of field components. That means that by paying and getting these projects going on the islands, we impacted in an indirect way around 200 families that were allowing to feed the participants, move them around, captains, drivers, during the length of this project. One of the pillars that we are very excited that this is working has to do with citizen science and democratizing science. And this photo here is the best example on how we achieve that. We have a fisherwoman and a farmer working together after they have collected seawater from their shores. They're filtering water through this pump and collecting filters for DNA extraction. They will go into the lab in the afternoon, extract eDNA from these filters, metabarcode it, and in the next few hours, they will be able to look into the results. And the empowering of the community by having this type of technology was the key success in our project. But to do that, we need to start building capacity locally. And one of the things that we encounter as our bigger was the gap between the knowledge in between the participants. So we had to develop a way to deliver equitable learning practices to reduce and remove the inequalities in access to knowledge and technology. Technology innovation obviously was the core of our work, and we use Oxford Nanopore as the technology that would allow us to reach our goals and also to match our logistic necessities, which is in the middle of the Pacific, on an island during the pandemic. And this work was done in collaboration with local scientists that help us to design some of the research projects. We also wanted to include the young and new generations of scientists on the Galapagos, where 2 of the students in this group, they were able to design their own honors and graduate student thesis used towards their degree. And finally, we also wanted to record the experiences, the hurdles, and how that information could feed back into the model and help us understand what are the things that we're learning and the things that we're doing correctly and the things that we could change. So in sum, This is the outcome of a hard work for over 74 locals over 3 islands that they spent countless hours in the field collecting water, soil samples, invertebrates, plants, and hours in the laboratory producing thousands of sequences of Galapagos species for the first time ever. And one of the outcomes thus far is that we have already identified several new species, including one species of fungi associated to an endemic plant on the Galapagos. So for the rest of the talk, what I'm going to present today is going to be 4 research projects that has the scientific background and which in conjunction with the community we have designed to answer questions in terms of cataloging biodiversity on the islands and how can we use nanopore technologies to support these type of research. So the first project that we started has to do with soil microbiomes. And I don't have to tell you, but the Galapagos is a living laboratory, which means that the information that we know about the origin of species above ground is very well understood. So we know how islands in isolation affected the generation of species like these giant tortoises there, or the geologic age of these islands affecting lineage formation and speciation. What was the remaining question is to which extent these above-ground mechanisms are also affecting the below-ground biodiversity. So we created the island microbiome, and in this project we have all these participants from 3 islands going to the field and collecting soil samples across elevational gradients, extracting DNA in each one of the islands where they already have implemented a laboratory, and including in that photo, participants in an active volcano to look for thermophilic bacteria in the lava on the island of Isabella. And just to give you a little taste of what we found in that POC blocks, we can see here, these are the 16S barcoding kit that was able to use. And we started looking already a pattern in which older islands seem to have lower biodiversity of microbiomes. A second project that we implemented with the collaboration of locals was a monitoring program to identify the community of sharks in places that are heavily visited by tourists. And these are your shark cafes. The idea here was to use the vessels of locals to go to these places and collect water, filter the water on the boat, and with that environmental DNA, able to identify using barcodes and CO1 in particular, the number of species that we could find at each point. So in each one of those horizontal bars is one of those shark cafes, and every color represents the number of reeds that match a given species. So this will give us a snapshot of the composition and diversity of shark species at a given point. And this could be a different and more effective way to monitor biodiversity in the future. We can start looking at real-time seasonality patterns, movement between islands, and even the discovery of new species. The Galapagos Genomes is a project that takes on the org1 platform, which is the de novo sequencing of species of conservation concern. So the idea here is to generate these genomes that could allow us to understand a little bit better what is the current conservation status of these species, looking at signatures in the population level that could hint us that there is a conservation need. We can also start understanding the geographic distribution of genetic diversity, identifying potential populations that could be used to translocate and in rewilding projects that are already in place on the islands in order to apply genetic rescue. In this case, we are targeting the Galapagos petrel, a critical endangered species, and the San Cristobal mockingbird. And the last project that I want to talk to you today has to do with using the capacity already built on the islands by different organizations that have already put forward big efforts to collect all these natural history museum and collections. So using the collections on the islands and using barcoding techniques, we wanted to identify and, first of all, define taxonomic boundaries of already described species, identify if any of these species are actually invasive, which are one of the biggest threats to the islands, and most interestingly, we could probably start also looking at the identification of new species. So with the Barcoding Galapagos project, we wanted to use already existing natural history collections to unveil hidden biodiversity on these islands. In this slide, I'm presenting the framework or the summary of what the Galapagos Genetic Barcode is about. And one of the aspects that I haven't touched much in the previous examples is that we are very excited in how we can, with this technology that has already been implemented on the islands, with the capacity that we have already built on the islands, to support the conservation of endangered species from poaching and illegal trade. You will think that the Galapagos is immune to these type of events, but they're not. They're more common than one wish they were. So the idea with this project is to help the authorities and organizations on the islands to identify swiftly species that can now be relocated back to the natural environments. In this case, there were 125 Galapagos giant tortoises seized in an airport. They did not know which one of the 15 species of of Galapagos tortoise species belong to. You can either wait 40 years for these tortoises to grow and identify them morphologically, or you can use barcode and move those specimens back to their original island. The other thing is that by the swift production of these information, legal repercussions are now going to be in place. And instead of waiting several months before we can get any identification if these species are under conservation status, this information can now be put in place and take action. So one of the final messages about my talk here is that the Galapagos Genetic Barcode is a model that can be implemented on other island nations or on areas in which the communities rely heavily on ecotourism or areas that are known to have high biodiversity. But most importantly, the Galapagos Genetic Barcode is a model that should and could be implemented in regions where communities understand the importance of preserving ecosystems and where technologies like nanopore could really help in decision-making and deciphering and understanding biodiversity to protect those environments, to help the livelihoods of the native communities there, and hopefully species around the planet. If you're interested to hear more or read more about our work, I invite you to use these two QR codes here. And I want to end by thanking an amazing group of collaborators from many parts of the world. I want to obviously be thankful to our three major funding organizations would allow us to employ locals for over a year and a half, to Nanopore Technologies for their continued support in generating all this genetic data on the islands during the pandemic, which was not an easy endeavor, and as many organizations, both locally and internationally, that support the implementation of this model on the islands. And a big thank you to all of you for your time.